my name is Riley Rafferty and today is April 25th. In this video, I'm going to explain how I carried out a DNA extraction using split peas. Before I begin the steps, I will briefly explain what DNA is and why it's important. Deoxyribonucleic acid, or DNA, is the molecule that carries almost all genetic information in all living organisms. It acts like a biological instruction manual, telling cells how to build proteins, how to divide, and how to function. Every trait is determined by the sequence of nucleotides in DNA. In this experiment, I extracted DNA so it could be seen by the human eye. To start the experiment, I added split peas, cold water, and salt to the blender. The salt is added because it helps break apart the cell structures and neutralize the charges on the negative DNA. By reducing these charges, the DNA strands are able to clump together rather than repel each other. This step sets the stage to cleanly separate the DNA later in the experiment. Next, I poured the blended mixture through the cheesecloth to help remove the solid pea material. What drips through is a greenish liquid that contains contents of the pea cell. This includes the proteins, membranes, starches, and the DNA that we're all trying to isolate. This filtrate is what we will be working with for the rest of the experiment. After straining, dish soap was added to the solution and stirred gently. The detergent dissolves the cell and nuclear membranes, which are made of lipids, because soap molecules are designed to dissolve lipids. By breaking down those membranes, the soap allowed the DNA to be released into the solution. It was then stirred very slowly so that the DNA strands would not be broken apart. The solution was then placed in the fridge for 10 minutes. Once chilled, the solution was poured into a test tube until the tube was about 1/3 of the way full. Cooling the solution helps slow down enzymes that might be used to degrade the DNA. Next, the pampin powder was added. Pampin is a protease enzyme that helps break down proteins, including the histone proteins that the DNA is wrapped tightly around. By digesting the proteins, the pampin helps release the DNA and prevents it from staying tangled. The mixture was stirred very gently. After that, the ice-cold alcohol was slowly poured down the side of the test tube. This created a separate alcohol layer at the top of the pea solution. The chilled alcohol caused the DNA to participate because DNA is not soluble in alcohol. As the alcohol mixed with the solution, the DNA began to clump together. The tube was allowed to sit for about a minute to let the process continue to run. At the interface between the two layers, I observed the DNA starting to form. It appeared as white, cloudy, stringy filaments. It had a sticky, rope-like texture and clumped together very easily. In my sample, the amount of DNA extracted was less than I had expected. I had anticipated seeing a larger mass of DNA. But this outcome is expected because only a little amount of the DNA was precipitated during the extraction process. I completed my DNA extraction. This experiment showed how simple chemical steps, like adding salt, detergent, enzymes, and cold alcohol, allowed me to isolate DNA from pea cells. Thank you, and I hope you enjoyed.